Green tea has been a staple drink across Asia for thousands of years. It boasts many health benefits thanks to a group of compounds known as polyphenols. What are polyphenols and what makes them good for us? Let's find out. Tea refers to the species Camellia sinensis and the plant originated in China. Typically, the leaves are harvested and pan-fried or roasted. Then it will be fermented to make oolong or black tea. Green tea, however, is not fermented. It contains the highest amount of polyphenols out of all types of tea and was traditionally consumed for its function as a diuretic, stimulant, blood sugar regulator, and many more. Polyphenols are compounds that have multiple phenol rings in their structures. They are known for their antioxidant properties. The main group of polyphenols in green tea is called the catechins. They all share the base form, which is catechin, and are all known antioxidants, in which EGCG is found to have the highest antioxidant power.
Before we go into further detail, it is important to understand what an antioxidant is. An antioxidant is a compound that inhibits oxidation by donating an electron to a free radical. This electron comes in the form of a hydrogen atom, and free radicals are highly reactive and like to react with other compounds to form more complex radicals. Antioxidants work by terminating this propagation step. An antioxidant's power is basically proportional to the number of hydroxyl groups that it has. As shown earlier, phenol contains a hydroxyl group, and since polyphenols have many phenol groups, they are strong antioxidants. Compare alpha tocopherol or vitamin E to a catechin. Catechins are much more potent antioxidants because they have more hydroxyl groups. There are actually many different ways that radicals can be generated. Outside of the human body, things like radiation and cigarette smoke are a source of radicals. More interestingly, some internal body processes also naturally generate radicals, like the immune system, certain enzymatic reactions, and internal Fenton's reaction. As for the general mechanism of how antioxidants work, after a radical has been generated, it usually doesn't last very long due to its reactive nature, and so it will look for something to react with. Radicals typically react with oxygen to form peroxyl radicals, indicated by ROO. Peroxyl radicals are not really stable molecules either, and they will too look for something to react with. They can react with antioxidants as indicated by the radical trapping reaction. The initial radical is quenched and now the antioxidant becomes the radical. As long as there is another radical species present indicated by X, the antioxidant radical can react with it to form a non-radical product. X in this case would most likely be another antioxidant radical.
Inflammation occurs when pro-inflammatory cytokines and reactive oxygen species activate transcription factors, which are then translocated into the nucleus and upregulate a variety of inflammatory genes. EGCG's anti-inflammatory actions include scavenging ROS and preventing translocation. An example would be in the study where mesangial cells were stimulated with LPS. Those which were treated with EGCG prior to stimulation had a significantly lower expression of inflammatory proteins such as INOS, as shown on the graph on the left, and nitrite, as seen in the graph on the right.
Regarding the relationship between DNA damage and the green tea polyphenol as antioxidant, there is a description calling this the antioxidant dilemma. In a couple of papers showing the green tea polyphenol such as EGCG are highly dose and environment dependent. For example, in a study in 2013, it is reported green tea polyphenol can protect against quadrivalent chromium-mediated DNA damage and apoptosis in vivo. It involves the typical mechanism as mentioned previously, that it can inhibit the oxidative stress by scavenging the free radicals and reactive oxygen species. In another study reported in 2007, the EGCG in solution may affect the DNA hypermethylation with a concentration of 50 times our regular plasma EGCG. The GCG can inhibit the DNA methyltransferase and also react with some methylation silence genes. However, another publication in 2013 reported EGCG in green tea solution can actually cause reductive damage to DNA. It is reported EGCG in green tea solution can be highly toxic against the human lung and skin normal cells, and it's even more toxic than hydroperoxide and CDDP, which is a typical carcinogen. As shown in the figure, a high concentration of antioxidant solution rich in weakly bound electrons and superoxide. These electrons in excited precursor state near DGMP nucleotide will form a transient molecular anion resonance that then leads to the molecular bound breaks in DNA, and eventually the single strand or double strand DNA will break. In this slide, a comprehensive model is shown. The author described the oxidative stress as a double-edged sword, which means the oxidative stress can be a factor of carcinogenesis, but it also reduces the risk of infection by promoting inflammation in some cases. Also, the reductive stress was described as a forgotten enemy. Previously, we only focused on the benefit of antioxidant in terms of protection from oxidative stress. However, too much antioxidant may push the equilibrium to the other side, where a strong reductive stress could also increase the risk of carcinogenesis. Overall, this model was conveying the concept of a balance between the intake of antioxidant and oxidative stress in our body.
Obesity remains a global health problem, so there are many researchers interested in the anti-obesity effects of green tea polyphenols. Epidemiological evidence and several randomized control studies have shown that green tea polyphenols, especially catechins, may affect multiple energy balance and result in decreasing of body weight, body fat, and waist circumference. Most of the studies focus on the EGCG's ability to increase the energy expenditure and fat metabolism. There also are some studies propose that EGCG may also suppress appetite and reduce nutrient absorption. Tina Ring's review paper summarized some human studies about EGCG and EGCG caffeine mixtures, effects on the energy expenditure and fat metabolisms. Most of the studies showed increase of energy expenditure in caffeine and EGCG mixture treatment. However, only a few studies compare EGCG caffeine mixture to equivalent amount of caffeine, and they are failure to show significant differences. On the other hand, these studies also suggested that EGCG may stimulate energy expenditure in a dose-dependent manner. Sympathetic nervous system plays a major role in the regulation of energy expenditure and lipolysis. EGCG could inhibit the COMT, the enzyme that degrades norepinephrine, thus prolonging the action of norepinephrine. Caffeine can inhibit a phosphodiesterase to decrease the degradation of CAMP and increase NE response. The appetite inhibition may be the result of increasing of fat oxidation and ATP levels. In addition, EGCG may also inhibit gastrointestinal enzymes, especially alpha amylase and alpha glucosidase, to reduce glucose absorption. Based on currently available literature, green tea polyphenols may also have a beneficial effect on total and LDL cholesterol levels. Animal studies have suggested that green tea polyphenol may reduce lipid absorption in intestines, promote fecal excretion of cholesterols, and inhibit enzymes involved in hepatic cholesterol synthesis. In human beings, Large epidemiological studies also suggest the efficacy of green tea polyphenol in reducing lipid levels. However, there is no statistically significant effect on the HDL cluster.
EGCG is a hydrophilic molecule, and therefore its efficacy can be quite low in lipophilic or biological systems. This is especially important when considering its cellular absorption in vivo. In order to enhance its bioavailability, EGCG might be lipophilized. In a study conducted by Zhang and Shahidi, EGCG was esterified, creating 3 ester derivatives, which will be referred to as compounds 1, 2, and 3. One part of their study involved testing how well EGCG and its derivatives can be incorporated into the core of liposomes. This process was used to estimate cellular uptake of those compounds, with the liposome's lipid bilayer modeling as a cellular membrane. The results, as seen in Table 2, show that compound 1 had the highest percentage of incorporation into liposomes, followed by compound 3, then compound 2, and EGCG having the lowest percentage. Generally, the trends show that the more lipophilic the compound, the higher the percentage of incorporation. Another concern regarding bioavailability of EGCG is its quick rate of degradation. A study showed that after incubating EGCG in simulated intestinal fluid of pH 7.4 for 1 hour, approximately 80% of the compound was degraded. A subsequent study concluded that encapsulating EGCG with chitosan nanoparticles can increase its stability and ultimately its oral absorption. The researchers measured the cumulative amount of EGCG that was transported across the mouse jejunum from the mucosa to serosa. When comparing the amount of encapsulated EGCG to the non-encapsulated ones, they found that absorption across the jejunum was increased after encapsulation. At 2 hours, there was a significant decrease in the amount of non-encapsulated EGCG being transported. This was attributed to the result of degradation. On the other hand, the amount of encapsulated EGCG continued to increase at 2 hours and beyond.
In the present, you don't have to look very hard in order to find green tea products. Most of these you can find in many supermarkets. There's the classic green tea leaves and green tea bags, such as Longjing tea leaves and Lipton tea bags. Matcha has been especially trendy in the past few years, so it's quite common to see matcha powder in stores. And in the natural health products section in supermarkets, one could easily find green tea extract in the form of pills. According to CFIA, there are several health claims manufacturers can make about green tea products used as food and only for drinking purposes. The 3 claims are quite similar, each relating to antioxidants, blood, and blood lipids. In order to make these claims, there are certain conditions that have to be met. For example, a cup of tea made with leaves, there must be at least 2 grams of tea leaves in 250 ml of water. This requirement must be considered when the manufacturer is creating instructions on how to make their tea. Manufacturers may also decide to warn consumers that they should not drink more than 9 cups of tea per day in order to avoid overconsumption of caffeine. Green tea extracts and natural health products can also have certain health claims. To make the claim that the product is a source of antioxidants for the maintenance of good health, a daily dose of the extracts need to contain up to 690 milligrams of catechins but under 150 milligrams of caffeine. To make the claim that the product is to be used with a program of reduced intake of dietary calories and increased physical activity to help in weight management, a daily dose of extract must contain 136 to 300 milligrams of EGCG, 75 to 150 milligrams of caffeine, and achieve an EGCG to caffeine ratio of 1.8 to 1 to 4 to 1.
In conclusion, consumption of green tea and green tea products may help with disease reduction and have some beneficial effects, but we may also see adverse effects such as DNA reductive damage in some circumstances. Catechins in normal green tea also have low bioavailability, so it wouldn't have much of an effect unless you take some type of green tea natural health product that increases that bioavailability. The overall recommendation is to not rely on green tea for good health. You can consume leisurely in moderation, But for a healthy human being, drinking too much green tea may do more harm than good. So seek medical assistance if you wish to take green tea or green tea products for more significant health benefits.